Well, thanks very much, John and Norma, for the opportunity to share a little bit of our work.
Mike.
So our lab really focuses on. Sorry, there's never a podium that it's. I'll dig in like this. Okay. Just kind of sit like this. Yeah, it's a lot easier. Okay. So my lab really focuses on spatial technologies and combined with artificial intelligence to try and develop both biomarkers for response to therapy and also to try and understand and interrogate the tumor immune microenvironment for new therapeutic vulnerabilities. And I have nothing to disclose, which is sort of PhD talk for the room that I am poor. So I think the secret's out. Everything is really moving towards spatial. There's a huge boom in spatial technologies, whether it's transcriptomics, whether it's proteomics, epigenomics, metabolomics in 2D and 3D. There's even 4D studies coming out, which are doing spatial over time. I wrote this review about a few months ago, and it's the first review I've ever written where all of the reviewer comments that came back saying, oh, you missed this paper and this paper and this paper were all bioRxiv or medRxiv papers. So unpublished work. So just a testament to how fast this field is really moving, not just from a technology standpoint, but also from an analysis standpoint. I think this has really been the bottleneck in the field in terms of having the right analysis tools to be able to interrogate these types of data sets. And so my lab spent a number of years trying to develop some sort of analysis tools that we can use for these various technologies. We started working on segmentation several years ago. And when we started this work, it was very clear that the tools that were available to properly segment cells were just not sufficient for the accuracy that you need to really make inferences from this type of technology. If you can imagine, if you cannot identify where every single individual cell is and you can't segment them, then your data is just going to be riddled with false positives. And so we teamed up with this Kaleem Siddiqui, who's at the Center for Intelligent Machines here at McGill, and this is work done by Elham Karimi and Morteza Rezanejad in my lab. And we spent a couple of years developing this segmentation approach. It's a bunch of fancy math. It's machine learning combined with classical computer vision. It's an algorithm we call Circle. It's available on our GitHub. We've since spent another 2 years developing a foundation model, and we have a second version of this that's going to be coming out very, very soon. And this will be publicly available. So if you are doing spatial stuff, I encourage you to look very closely at how your cells are being segmented. And please, if you want to check out our tools, we make them all available for you to use. And this is just a sort of qualitative example of how our tool performs against a number of different, classical computer vision on the right and machine learning tools that are popular on the left. Okay, so in addition to cell segmentation, especially in the proteomics, spatial proteomics field, cell type assignment has also been a bit of a challenge as well. So we developed an additional tool. Again, this is on our GitHub. All of it's available. It's sort of a pixel level ranked hierarchical approach. And essentially what you can do is you can take any sort of raw images, which you can see on the left, and you can then segment them and digitally assign them to any of, in this case, 20 different immune lineages. And if it's tough to see that one, this is just a sexy picture of an appendix that's really nicely segmented and assigned to various different immune components. So once you have the ability to run these technologies, to do the segmentation and cell type assignment, you can start to ask questions. We're extremely fortunate in our group to collaborate with both John and Pierre and with Philippe Joubert at University of Laval. And so together we've been able to curate over 800 tumors that we've profiled with the various technologies that we have at the Goodman Cancer Institute where I work. And when you do these types of data sets, there's really 3 types of analysis that you can do. So the first one, which is sort of the low-hanging fruit, the kind of the data that it spits off, and that's just frequencies of cells. But you can start to ask questions about, is there some sort of correlates with the frequency of cells and any sort of clinical variable you're interested in. So in this case, a couple of things that pop out. There's a big sort of, I don't know what that color is, but you can see the late-stage patients are really enriched in neutrophils, or patients that are really enriched in B cells tend to do really well by survival. So that's sort of the first tier of analysis, is just looking at frequency of cells, but that's not really why we do spatial. This thing doesn't work anymore. Okay. So you want to do spatial because you want to know how the cells are organized in the tissue. And so there's 2 ways of looking at this. One of them is just looking at pairwise interactions, so how 2 cells spatially interact with each other. And this is just to give you an example of two tumors. And if you want to look at the interaction between cancer cells and CD8 T cells, which are white and green, the little blow-ups in the middle show you that in the tumor on the left here, what you can see is that the CD8 T cells are much more interspersed within those white tumor cells, whereas the tumor on the right, they're much more immune excluded, so they're not really physically getting near each other. And you can see that qualitatively, but if you want to quantitate that, we've sort of made an approach where you can absolutely qualitate this by asking within an image how many of those T cells are touching a cancer cell or within a certain distance. You can define it as 4 or 6 pixels. And then we randomly scramble the image with maintaining the proportions, and we ask by random chance, what is the likelihood of these two interacting? We permeate this about 50,000 times, and then we get an absolute quantification of whether or not two cells are likely to interact with each other or avoid each other. And you can do that by comparing cohorts as well. And again, all of the code to run that type of analysis is available on our GitHub. Man, this thing's bad. Can you go next slide? Okay, this is the next slide. So there's a number of tools beyond pairwise analysis to look at broader cellular neighborhoods or cellular communities. This is just one example of a tool that we helped build with Giovanni Cirello's lab in Switzerland. It's called Cell Charter. If you're interested in knowing how it works, there's a really beautiful little in silico talk by Marco, who's the student who led this project. But regardless of the tool that you want to use, the idea here is that you want to look at not just pairwise interactions, but how the cell is distributed in terms of neighborhoods or networks or communities or whatever you want to call them. And then you want to look at how those distributions correlate with any clinical variables, whether it's response to therapy, whether it's survival. And it gives you a different level of resolution in terms of how the tissue is organized and how that may affect the biology of that patient. And so we've used these tools to do a number of different studies over the last couple of years, starting with a paper that John and Pierre and I published, sort of the largest proteomic spatially resolved atlas of lung adenocarcinoma with over 500 patient tumors. We did a small study that we published in JITSI looking at recurrent tumors that were treated with IO. More recently, we've been able to curate driver gene data. So in a cohort of about 200 patients, we'll be able to correlate the immune microenvironment and how it's spatially resolved with specific driver genes to show exactly what those contributions of those individual drivers are to the tumor immune microenvironment. We have a paper out for review, John and I, that's looking at the difference between adeno and squames. This is a clinically matched cohort, so we use propensity matching to clinically match these cohorts so you can really start to dissect without having confounding variables to look at the difference between the immune landscape and how it's spatially resolved in adenos and squames. And then we have another paper looking really deeply at smoking and the effects of smoking, in addition to a number of other cohorts that we've looked at, including STAS, these multiple sclerosis lung adenocarcinomas, neuroendocrine tumors, Saber Bridge, and of course the one that we're really, really focused on and interested in that we've been doing transcriptomics, proteomics, et cetera, is these early stage neoad IO patients. Okay, so beyond the sort of diving into the microenvironment and using these tools to sort of ask about the biology of these tumors, what we're really focused on is using AI and how AI can predict clinical outcomes with some of these tech. And so the first example of where we showed success in this space was looking at stage 1 patients. And so we wanted to ask the question simply, after surgical resection, can we use some sort of an AI model combined with a spatial omic technology to predict recurrence in these patients? And so we took 285 stage 1 patients and we trained a model. And then we took an additional 120 cores, and we asked a simple question. We knew this was retrospective tissue data, and we said, can we predict whether or not these patients are going to progress or not? And the answer is we could do it at about 95% accuracy, which is pretty amazing considering what you've heard about using liquid biopsies and whatnot. And this is from a 1 millimeter piece of tissue, smaller than a routine core biopsy. Further to this, we used a transfer learning approach, which we started this a couple of years ago before like ChatGPT and all these large language models became cool. So we feel like we're kind of trendsetters in this space, if I might say so myself. But now that we're using some of the more recent tools, there's a huge surge in tools and different type of approaches, which we can talk about afterwards. But we've been able to re-look at this data, and we can do it at over 99% accuracy. So think of the potential treatment response. If we can get the right cohorts, apples to apples cohorts, I think this is a way in which we can look for amazing biomarkers or tools that can start to triage patients onto whether they're likely to respond or not responding to specific therapies and whether or not they're going to recur or not. We also have sort of, it was mentioned earlier about how, H&Es are, $5 a shot, and we're very cognizant of the fact of what's actually clinically implementable. Sometimes these big spatial data sets aren't very implementable in the clinic. We have been able to titer down instead of a 40 gene panel or 40 antibody panel down to about 3 antibodies, which we can do in sort of the mid-90s. But we wanted to do something even better. And I was going to talk about prediction numbers, but I think I've never shown this before, but I think a picture is worth 1,000 words. So we've trained one of these models, okay, on different modalities. Okay, so we made these large models, whether it's on radiographs or H&Es or IHCs, and this is an example of a model we've trained on just a 1 millimeter. This is just an example of one core 1 millimeter tumor. And in the middle, this model's never seen this image before. Never, ever. And I gave it this. 80% of the image is completely blacked out. And I said, what do you think this thing is supposed to look like? And this is what it does. Again, never seen this before. I black out 80% of the image. It tells you exactly what what it looks like. Okay? And this isn't even a huge model. We have the potential as a community to build this type of a model off of thousands and thousands. We do millions of little bits of H&Es, but we could do it off of, collectively thousands upon thousands of tens of thousands of patients. Okay? It's not just H&Es. This is an example of seven slices from a CT scan. So again, on the left is just they've never seen this CT scan before. We block out about 80% of the image in the middle. And on the right, you'll see what it renders. And it'll keep going, and it's very accurate. If you don't believe it in 2D, check it out in 3D. So again, this is a 3T CT scan. For purposes of like the size of the image, I just did it in this format. The middle, again, blocked out 75% of the image, and look on the right of what this image predicts this CT scan to look like. And so these are foundation models, okay? This is the backbone of how we actually do learning. So you train a model like this, then you can say, these patients are responders and these weren't responders by CT or by H&E or by IHC or whatnot. You can say, these patients were good candidates for surgery or these candidates were poor candidates for surgery. And if you had enough cases, and I know collectively in this room there are enough cases, we could definitely start to answer some of the challenges that were outlined a little bit earlier in terms of, is it really the imaging modalities that we have available? Do we need to go to liquid biopsy? Is it just H&Es that work? I think as a first pass, we have these models available, we have these tools, we just need enough samples to be able to build them, to be able to ask those questions. And I think what's really most exciting for me is that, this is an example of a picture of just for explanation purposes, this is my dog. My dog's name's Dappy. Because of the blue eyes. Obviously, we crowdsourced the name, and my friends are all science nerds, so they said Dappy. This is my son playing with my Snapchat on my phone. So, but for the sake of this, it's just to explain to you that when you train these models, okay, you showed a picture of a dog, and then you showed a picture of a cat. You say, this is a dog, this is a cat, this is a dog, this is a cat, this is a dog, this is a cat. And then you showed a picture of a dog, and it'll say, this is a dog, with like 100% accuracy. Okay, it's extremely accurate, these models. The question is, what is it looking at? If you think about, from our perspective, when I look at the picture, I'm like, obviously it's a dog and it's a cat, but what makes that a dog and what makes that a cat? And what these models are really good at is distilling down to the very smallest differences that can discern two things. So for example, in this case, maybe they're not looking at the entire image, maybe they're just looking at the pupil. And the shape of the pupil is all you really need to differentiate these two. So we've created a pipeline, in this case, where we can go ahead and use something called guided backpropagation. So on every single tumor core, when we have an accurate prediction of whether a patient responds to therapy or progresses, we can go back and we can actually get what looks like almost a heat map on that image or on that core, so we can identify the very specific small region on those tumors that's actually being used for the prediction. And the hope is that those regions are going to allow us to learn what is most important for response to therapy, for progression, et cetera, and we can learn from the biology in those spaces. So I think there's incredible opportunity here. We're doing this again, of course, in the neoadjuvant chemo IO case. We're always looking for more samples. We want really clean, beautiful data sets to do this type of stuff. We're really excited about the potential to hopefully help clinicians in terms of decision making, and then also for us to learn for new therapeutics in the future. So with that, I'd like to thank my lab, all of our collaborators, our funding sources, the patients and their families. And I don't know how much time I have, but I'd be happy to take one or two quick questions.
Really great talk. You said at the end that it will tell you specific parts of the slide to focus on to see what it is that's differentiating. Does it also tell you which characteristics, or does it just say, look at the left upper corner and that's what we found interesting?
It'll show you the region, but the example that I just gave you there was an example of tumors that we spatially profiled with like 40 channels for proteomics. So we can go through every single channel and we can get what is the contribution of each antibody in each channel, what are the cell types that exist, and what is the activation state of those cells in those specific regions. And then on top of that, you can ask not only what are present, but what is the communities that are involved there. So how are they spatially organized, and is that important for outcomes? And you can do it across every single prediction, and if you have cohorts of 500 patients, you start to be able to really distill down what are the individual things that are contributing in that space.
Thank you for your talk. Do you think that we could teach AI to tell us from a slide of a biopsy that there might be tumor heterogeneity in another part of the tumor that was not biopsied?
It's a good question. This is always the biggest thing that comes up is tumor heterogeneity. We, in our initial predictions for early stage, what was really, I thought was the most shocking thing is that I had Pierre just sort of randomly take two distinct areas of the tumor, so 2 cores per tumor. When you look at them on H&E or you look at them by multiplex, they look completely different, right? The cells that exist there are totally different. We still had, I think, 97 point something concurrence in the guess or the prediction, even though they look totally different. So we don't exactly know why it is, but I think if we had a bunch of examples of that where you had multiple samplings from the same tumor and you were able to give it heterogeneity based on the makeups of each of those points, I don't see why not. I just think that there's incredible power in these technologies. I think, the way we need to start thinking about it in terms of, the question was asked about what's happening 30 years from now. I think this is definitely going to be a big, big, big part of that story. And, I think pathologists, and I always feel bad when I say something along this with pathologists, but, they spent 15 years, and it's a high, it was discussed earlier, that's a really high bar to train people to be able to look at these tumors. But when you compare the ability of some sort of AI model like this, it's the difference between a supercomputer doing multiple calculations at a time and, sort of us counting on our fingers because our eyes can only look at so many different features at one given time.
So I guess you didn't go into that detail, but you could probably train your model to predict IHC and molecular features of that tumor as well.
Absolutely. So anyone that's interested and has cohorts and has questions, we're open and we're available to build these models and work with you with those tools.
Thanks so much. All right.